Today, I'd like to welcome everybody to our 12th webinar and our two presenters that we're very fortunate to have today, Dr. Carol Boss and Dr. Jorge Nieva. Carol is a longtime friend of this group and has presented. This is her third time for presenting. She has a PhD in biomedical engineering and has a professional life as well as a very, very full and wide, broad advocate volunteer life. She's in actually meeting the needs of almost all the stakeholders that are involved, from academia to government to patients on an IRB to advocate groups consulting for CISN. So winning awards across all of that spectrum because of her involvement and expertise. So thank you so much, Carol, for your presentation today and for your friendship and involvement in all aspects of advocacy, and for bringing Dr. Nieva along today, because his expertise will be greatly appreciated by the group. So Dr. Nieva, a little bit about his background. He graduated from University of California, Irvine. He had training in internal medicine, oncology and hematology at Scripps Clinic. He worked there for quite a while. He was involved and pioneered a new technology for the detection of cancer cells in the peripheral blood and discoveries related to the fundamental mechanisms of the immune system. So we've talked about that a little bit in other talks, and now we have an expert in that field. So that's going to be really interesting to hear from you, Dr. Nieva. So thank you. He was then recruited to Billings Clinic in Montana and was a department chair and program leader there, and involved with the multidisciplinary lung cancer and head and neck cancer clinics. Then moved back to California and is at USC now. He's won many awards, including ASCO for Excellence in Clinical Trials from the NCI, and also Outstanding Teacher Award from Scripps Clinic. So thank you, Dr. Nieva, and we'll hear from you. Carol is going to go first, and then he will follow up. So why don't we move forward? We're going to kind of structure it the same as we always do. We'll have breaks, and Carol will let you know when that happens. And please type your question in before that point so that I can see if you have one, and then I'll unmute you. Our mission as a nonprofit is to bring all the stakeholders together and to deliver information both to advocates, the community, and to researchers and professionals. And so that's what this webinar series is about. And here's where we start. And Carol, please feel free to take over.
Sure, thank you very much. This is a topic that is near and dear to my heart, so I'm going to enjoy it, and I hope that all of you do as well. I'm going to try to talk pretty quickly so that we can get to the heart of it, which is Dr. Nieva's work. So let's see, is the slide that you can see right now learning objectives?
Yes.
Okay. So what I would like for you to know by the end of this session is the fields that are included in the physical sciences, what we mean by the term physical sciences in oncology, what the National Cancer Institute has done in terms of developing the field of physical science oncology, how advocates can be involved in this area, and then Dr. Nieva will give us a wonderful example of how the physical sciences increase our understanding of cancer. Next slide. So what we're going to do is we're going to talk about what the physical sciences are, just very briefly talk about the physical sciences in medicine, how transdisciplinary teams are developed to study cancer. Again, we're going to talk about how advocates are included and end up with some case studies. Next slide. So first, some quick background information for us. Next slide. So, a 21st century approach to cancer research. As we know, cancer is extraordinarily complex. We need new approaches, we need collaboration between all kinds of researchers, and we need interdisciplinary and transdisciplinary teams. So that's how we're going to win the war on cancer. So these advances in the physical sciences are helping us unravel all these complex aspects of cancer, creating new opportunities for prevention, detection, and treatment of disease. Next slide. So which scientists are going to be involved? We're going to have biologists, clinicians, and physician scientists, of course, but also physical scientists. So I get the question all the time, what are the physical sciences? So as you can see from the slide, they include everything from chemistry, physics, computer science, mathematics, and engineering. Next slide. So one of the reasons why I'm so excited about the physical sciences in oncology is because by training I am a biomedical engineer. So I want to talk a little bit about what biomedical engineering is. Wikipedia actually has a really good definition. So biomedical engineering is a vital part of the physical sciences and oncology effort. Biomedical engineering, in its simplest terms, is engineering in medicine. It combines the design and problem-solving skills of engineering with medical and biological sciences to advance healthcare. The graphic there, I'm not sure if you can really see it, but if you look around the circle, you can see all the different aspects of what they call bioengineering or biomedical engineering. Can include anything from mechanical engineering to physics to computer science to math. Next slide. So another term that we're going to be using is convergent science. So what is convergent science? This is actually a pretty good graphic. It describes, or it gives us the idea that when we talk about convergent thinking, what we're talking about is that we're going to have a lot of different types of scientists who are all going to be working together on cancer. And that's how we're going to achieve the ultimate goal. So when you focus in on a problem, that's what we call convergent science. Next slide. So this is an easy way to think about what convergent science is. We're focusing in on the problem, and we're doing that in convergent science by merging distinct technologies, industries, tools, disciplines, or devices into a unified whole. We're creating new pathways and new opportunities. So convergence relies on a new integrated approach to solving problems that are too complex for any single discipline. Next slide. So the history of physical sciences and oncology. I happen to be really interested in the history of medicine, but I will try very hard not to spend too much time on this. Next slide. So the physical sciences have been important from the very first days of medicine. This illustration gives you an overview of some ways in which physics have impacted medicine. Everything from the invention of the microscope to the discovery of X-rays to the advent of nanomedicine. Physical sciences have revolutionized the way that we understand, we diagnose, and we treat disease. Next slide. So this is actually a page from a book that was just published in November of 2015. The book elaborates on 6 topics that were identified to have the greatest potential to advance our understanding and treatment of cancer. So they have one section that's on cell mechanics, and they describe just one of the ways in which the physical sciences have changed the way we understand biology and physiology. So that the example that they give here in this particular section talks about the first microscopes. For example, in the 1700s, the developer of the microscope observed cilia, and this led to the characterization of cytoplasmic viscosity, which is actually, a technical term, but it's very important in understanding cell behavior. Next slide. So, what are the physical sciences in oncology today? Next slide. So in 2009, the NCI launched their Physical Sciences Oncology program. There were 12 centers that were established, and these centers investigated complex and challenging questions in cancer research from a physical sciences perspective. So this was a 5-year program. There were 150 main investigators that were from the fields of physics, math, chemistry, engineering, cancer biology, clinical oncology, and there were 110 institutions which were ultimately involved. And Dr. Nieva was part of that effort. Next slide. So there were 12 of these centers, and they were all over the country. You can see from the map. The focus was on 4 areas: physics of cancer, deconvoluting complexity, evolution and evolutionary thinking, and information coding and decoding. And the little image right up there at the very top on the right actually was from the first newsletter, which was produced as part of the PSOCs. So it was a 5-year program. The program has been renewed, so we're in round 2 right now, or phase 2. The PSOC moved within the NCI, and they're now under the Division of Cancer Biology. So this new initiative seeks to establish research projects that bring together cancer biologists and oncologists with scientists from physics, math, chemistry, and engineering to address some of the major questions and barriers in cancer research. Next slide. So right now, these are the physical sciences oncology centers here in the phase 2. So it's Columbia, Dana-Farber, Moffitt, Northwestern, and the University of Pennsylvania. And you can go to the NCI website. It's physics.cancer.gov, and you can get much more information about the NCI program. You can see what they're working on. There's links to articles and to the individual centers. Next slide. The NCI has also funded specific projects, and these projects are at Harvard, MIT, the University of California, Berkeley, the University of Utah, and Vanderbilt. So one of the things about the PSC was when I first heard about the program from another advocate, actually, she commented that I needed to get in touch with the NCI because this was a perfect project for me. And I did. I actually emailed the NCI. It was right after the program had started. They wanted to involve the patient voice in what they were doing, and I ended up becoming the lead advocate for the program. So for 5 years, I got to see the projects that they were working on and to make sure that the patient voice, that advocates were included at all of the centers. We didn't quite get to all 12, but we definitely made headroads, and I'm very proud of our efforts. Next slide. So in terms of what the advocates did within the PSOCs, probably I can best explain it by referring you to an article that was published in 2013 by Susan Sampson, was the lead writer. So advocacy was included from the very first in the PSOCs. This article details what the Bay Area PSOC did within their center in terms of advocates. So there's some very important statements in the article that I just wanted to call to your attention. So within the PSOC, there was the belief that the convergence of disciplines was what was going to permit the emergence of these new scientific principles and opportunities. She went on to write, and this article was actually written, it was a combination of advocates and researchers that were involved in the effort. And in full disclosure, I was also an author on this article. So advocates were a vital catalyst here. They synthesized, prioritized, and gave voice to what was happening. So advocates, as I think all of us can argue, advocates need to be and were in fact included from the initial and final stages of the research, development, and strategy. There was a bi-directional stakeholder engagement, and we were involved in reducing the cultural barriers that impair communication. One of the difficult things about being an advocate in this area is that it's a very complicated area, and it's very hard to explain sometimes to people who aren't in a technical field. But you don't have to be in a technical field to be interested in what's going on in the physical sciences and oncology. And we'll talk about that a little bit more later. Next slide. So here's some graphics that were from that article that I think really give us some idea on how advocates can be involved in this area of science. So you have physical sciences and you have oncology, and advocacy is really a bridge between the two. Advocates can give the physical scientist a real feel for who's going to be influenced by their research, because oftentimes the physical scientists don't deal directly with patients. You know, they're not in the clinics. So we add that real world focus and connection that really impacts their research and lets them know how important what they're doing really is. So advocates, if you look on the right side of the screen there, that graphic, advocates are involved in everything from metrics and measures, you know, on to how are we going to handle some of these thorny issues? How are we going to tackle issues that some of the researchers might not have thought of? Next slide. So there were a lot of advances that came out of this particular program, but there's a lot of researchers who are working in this space who are not part of the PSOC. So I just want you to realize that this really is a quite important and burgeoning area of science. So one of them, so I've just put 3 examples here. The example on the right is talking about a fluid biopsy, and Dr. Nieva is going to talk to us a little bit more about that. Then you can see the Nature Nanotechnology cover. Nanomedicine is physical sciences and oncology. And then on the far left, that article was actually just published, and it was published, the last author is actually Paul Macklin, who's a mathematician. And what his lab does, and I just wanted to talk a little bit about, you know, an example of how physical scientists are working in cancer. So what his lab does is it works to develop and validate models of cancer in individual patients. So they have tightly integrated teams that are clinicians, modelers, and biologists, and they develop computational tools that one day will help improve clinical planning. One of the things that they're working on, and that this particular article details, is they're working on a digital cell line. So this is a computer model that will have physical or describe physical and behavioral characteristics and the microenvironmental conditions. So this will help us understand cellular behavior in cancer, for example. Next slide. So one of the things that I was asked to talk about was what was one of the important developments that came out of the PSOC. And one of the things that did arise was the fact that it was discovered, or determined, I guess, that people who were working in this space, the researchers who were working in this space, they were publishing in all kinds of different journals because there was no one home for this type of research. So the Institute of Physics, go ahead and next slide, please. The Institute of Physics publishes a lot of different types of physics journals, and they decided that what they really wanted to do was to establish a journal that was focused on the physical sciences in oncology. So this new journal, which is called Convergent Science Physical Oncology, joined their stable of publications. And that's what we're going to talk about for probably the next 5 minutes, and then we'll move on to Dr. Nieva. So next slide. So this new journal is online only at this point. It's been around for about a little less than a year. So if you look at one of the comments by Peter Kuhn, who's one of the founding editors, I think it's a really wonderful statement, because one of the things that he comments on is, he said, when you work across the disciplines of medicine, biology, physics, and engineering, what you can do is you can really make new breakthroughs that can improve the human condition. But his last statement there is the one that I really want to call your attention to. Joint authorship of projects requires and indeed deserves joint editors, joint reviews, and joint readership. This is a journal that's really doing things differently. So I think the first thing that you'll notice is that there were 4 founding editors. This really is quite important, because one of the founding editors is me. So I'm a biomedical engineer, but first and foremost, I'm an advocate. So when the journal was founded, it was very, very important that there be representatives from the different fields that are involved in this type of research. So Dr. Kuhn is a physicist, Kelly Bethel is a pathologist, Dr. Nieva, who's going to be talking to us in a minute, is a practicing clinical oncologist, and then I'm the patient representative, of the 4 founding editors. Next slide. So one of the reasons I was so excited about this journal is that advocates really are an integral part. So not only do we have an advocate as a founding editor, but we have advocates who are members of the editorial board, as you can see by the list on the right. They're full members of the editorial board. Also, advocates can be authors, advocates serve as peer reviewers, and of course advocates can be readers. So CSPO includes patient-oriented science and perspectives, and also patient-authored publications. So I would encourage all of you to check out CSPO. I really want advocate readership, and I want advocate involvement. And please don't be frightened away if you're not that tech savvy, because what we need is the patient voice in all areas. And I think that most of you have been involved in grant reviews, for example, and you know that you're not, as an advocate reviewer, you're not expected to be an expert in the science, and that's the same case here. What you're an expert in is your experience as a patient. So that's what you're bringing. That's what you're bringing to the table. Next slide. So there's different types of articles that are within CSPO, and one of the very exciting types of articles that we have is called outcomes. So these are very patient-focused articles. So what we do with the outcomes is some of the research articles that are submitted are actually identified as being particularly suitable for a commentary that would describe how this particular research would really be important to the patient community. So then someone from the patient community, an advocate, is asked to write a short piece that describes the patient perspective. How does the patient community think about this work? We had our first one published. So we've had one issue published of CSPO, and we're working on our second one at this point. So on this particular slide, the scientific article is on the left, and so it was rapid changes in circulating tumor cells following anti-angiogenic therapy. And then on the right is actually the commentary by the advocates. So I was I was happy to give a short description of what I thought was going to be the impact of this particular work, along with Susan Sampson, who was the first author on this. So I just want to close my section of the talk with this, this quote, which is by David Ross Brower, who was a prominent American environmentalist who died in the year 2000. And this kind of sums up, I guess, how I think about the physical sciences. So understanding how DNA transmit all it knows about cancer, physics, dreaming, and love will keep man searching for some time. I hope it's not some time for us to figure out cancer, but I know it's going to keep us busy for a while. So anyway, that's the end of my section. And if we want, Peggy, we can hold it till after Dr. Nieva's part, because I want to make sure that we have enough time for him.
That sounds good. That sounds good. Okay, Dr. Nieva.
Hello, everybody. Hope you can all hear and see me. My name is George Nieva, and I'm basically Carol's helper on science, physical ecology. And I'm going to talk to you a little bit today about some examples of collaborations and convergence that lets clinicians and physical scientists work together. Next slide. So there's two things that you need in order to have successful convergence. You need a clinician with a problem, and you need a physical scientist with a technique. And probably the best example recently was when a professor of engineering found that, you know, if I put water or fat in a magnetic field, I can get the hydrogens to spin in different directions, and I can detect that spin. And you might think, well, gee, why does that matter? But that got him the Nobel Prize in Medicine for the invention of MRI scanning. So the physical scientist I've worked with for the last dozen or so years is a person by the name of Peter Kuhn, who's a theoretical physicist, and who really got his start in physics and medicine doing X-ray crystallography, basically taking pictures of very, very small objects. Next slide. And I think the key aspect, and for this relationship to work, really the two people need to understand each other's technology and need to understand the limitations of each other's technology and the limitations of the clinical problem. So for example, when I first met Peter, he said, I've got a rare cell detector, and it was my job as the clinician to really understand really what are the limitations of your rare cell detector, what can it do, what can't it do, and how do we find a clinical problem that we can work on. And then I think a good clinician will go in with the approach that it doesn't necessarily have to be my problem. The tool may be best served fixing someone else's problem, in which case the clinician should be willing to put the physicist in contact with the right people. Next slide. So I'm going to go over some case studies. I'm going to talk about technologies for fluid biopsies and circulating tumor cells. We'll talk about mathematical modeling of cancer. I'm going to talk about gene transfer, and I'm going to talk about analysis of movement. Next slide. So about a dozen years ago, I was brought into a room and I was told that the folks at Xerox PARC, which are the people who make laser printers, took a fiber bundle that was used for laser printing and could find rare events with it, could find rare cells in the circulation. And what they really wanted to go after is find fetal cells in the maternal circulation. And I said, well, that's an interesting problem, but wouldn't it be better to find cancer cells in the circulation? And so we worked on that for a number of years. And we spent, I would say, about 4 years working with the company, and then found that, you know, that may not be the best way to do things. And one of the challenges we experienced was that the engineers and the clinicians became somewhat disconnected from each other in terms of goals. And there were some problems with the technology. And the first thing that we had to do was recognize that just because we had an interesting tool didn't mean that that tool was the solution to our problem. So I had to, at the time, write this very embarrassing letter to my NCI program officer and say, you know, I wrote this grant to get all these funds to develop this tool, and my tool doesn't really work. And so we had to develop a new tool. Next slide. And one of the things we had to overcome was the NIH syndrome. And NIH doesn't stand for National Institutes of Health. NIH stands for Not Invented Here. And what we really needed to recognize was just because it wasn't our invention, it didn't mean that we couldn't actually approach the problem using a different tool. So we went back to another tool called an automated digital microscope, which had been around for a long time, but we thought had never been applied the right way and hadn't been applied with the right operational engineering processes to make it a good cancer detection technology. And so that's what we then did, is we developed this automated digital microscope, and we found we could find these red cancer cells that you can see on the slide here with really quite good resolution. Next slide. And, you know, serendipitously, we found that we actually had something that was very exciting. We had a tool that could not only detect cancer cells, but once it detected the cancer cells, we could actually store the cancer cells in frozen form. And what that really opened up the world to us was the amount of secondary science that we could actually do on those cells. We didn't have to define all of our experiments beforehand and then accrue the patients one at a time to get those experiments done. We could actually just accrue the patients as we were moving along, and then as the science advanced and new discoveries were made, we could then apply new discoveries to the specimens we had already collected. And that's something nobody else in the field could really do, and made this actually a very popular technique with pharma. And now this technology is used by Genentech and Pfizer and Abbott and many of the other big pharma companies in order to do science on circulating tumor cells. Next slide. And that's let us now move into the era of what we call the fluid biopsy. And this we think is very exciting technology. This is technology that lets us not just find cancer cells in the circulation, but characterize them. So it's not just about counting the number of cells, it's about understanding what genes and what proteins are on those cells so that we can give patients better information about the status of their cancer. Next slide. So this, what you see here, is a cluster of cancer cells that we've imaged. This is from one of my patients, and it shows enough cancer cells there. You can count 1, 2, 3, 4, 5, 6, 7, 8 cancer cells, all in a cluster. And once we analyze those cells computationally and genomically, we can tell what kind of cancer this person has, what molecular markers it contains, and we can say something about their prognosis. Next slide. So we then started to think about, can we make this better? Once we've found these cancer cell clusters, can we really use it to diagnose cancer? And one of the things that we recognized is that a diagnosis is really about probabilities. What is the likelihood that an event has occurred? And so we said, well, let's give you this example. If you want to diagnose somebody with malaria, if you know nothing more about them than they live on the Ivory Coast and you say they have malaria, you'll be right 5% of the time. If you say that, if you know that they have a fever, so you have a thermometer and a GPS, so you know where they are and you know their temperature, you'll be right 30% of the time. Well, if you also know that their blood cells are being destroyed through the process of hemolysis, and all three of those things are present, you'll be right 80% of the time. And then if you find the actual parasite in their blood, you're right 95% of the time. You're still not right 100% of the time. There could be other reasons that they're not feeling well. So when you think about a situation like the diagnosis of malaria, what you see there is that there's disparate data elements that are used to make the diagnosis, right? You need a map or a GPS, you need a thermometer, you need a blood smear, and you need some chemistry analysis to know if the red cells are being destroyed. So we took that same approach and again overcame the non-invented here syndrome to say, well, how can we put disparate data elements together to get at a diagnosis? Next slide. So what we did is say, well, it's really not all about our technique. It's not all about our technology. And one of the things that you have to do to make these collaborations work is you have to think outside your field, and you have to be willing to check your ego and say, well, maybe it's about whether or not the person's a smoker to diagnose lung cancer, for example. Maybe it's about how many cancer cells we find in the circulation, but maybe it's also about how old they are, and maybe it's also about what their PET scanner looks like. So we combine the technique with PET scanning, next slide, and came up with curves that now can be very effective at diagnosing lung cancer if we take into account all these different disparate data elements. Next slide. And so we then made a website. We made a graph where you could just punch in numbers. How hot is a nodule on a PET scan? How big is it? How old are they? A few historical elements, and then whether or not our fluid biopsy test was positive or not. And by doing that, we can come up with a more accurate probabilistic diagnosis of lung cancer than you can if you didn't have all those data elements. So what we really wanted to do here was not just think about our biologic technique, our cancer detection technique, but also some simple Bayesian mathematics, which we think is also part of the physical sciences. Next slide. Let's go one more slide. So here's a real example. This is one of my patients, and this is a nice lady with breast cancer. And what we did was found her cancer cells in the bloodstream, and actually then took a fluid biopsy and then turned it over to another collaborator who created a computational method for calculating the values of every element in the genome from a single cell. Next slide. So what we were able to do is now measure a whole genome on that single patient from a single cell and know which part of her chromosomes were amplified, which part of her chromosomes were deleted. Next slide. We could identify which genes were involved, but more importantly, we also identified that she didn't have just one genomic profile to her cancer. She didn't have a single cancer clone. She had multiple cancer clones that were genomically different from one another. And so here we now have to lean on our evolutionary biology colleagues and the science and mathematics of evolutionary theory and population selection to better understand exactly how these different populations were going to be affected by different treatments. Next slide. Because she didn't actually have 2 clones, she had 3 clones. And she had a 3rd clone that had 2 other amplifications in her DNA that the first 2 clones didn't have. Next slide. And these had unique genomic signatures. So we actually went through and analyzed each cell individually and found that most of the cells were in clone 2. There were some cells in clone 3 and some cells in clone 1, and what you see in this graphic is that each column represents an individual cell that we were able to do a whole genome on. And we really, couldn't have done that without some very powerful mathematical tools that can find a very small signal, from a single strand of DNA in one cell. Next slide. And then we also even did some very trivial things. We found that to ship our tubes of blood from one location to another, I was working in Montana at the time, where the temperatures could get as low as minus 20 in the wintertime. And so we were shipping our materials in these insulated green boxes. And then we worked with physical scientists to do something very trivial, to find temperature stabilization technologies that let us cut our shipping costs by a quarter. Again, same group of people, physician, physical scientist, but basically leveraging across disciplines to find solutions to problems. And whether it be a simple problem, like it costs us $100 to send a sample from Montana to San Diego and get it there by 10 a.m., to more important problems like, how do we determine how treatment is affecting the selection pressure on Mrs. Smith's cancer. Next slide. So we also did some work with mathematics and modeling. And the thing you need to understand about modeling is when you build a mathematical model, a representation of a disease, they're never perfect, and you have to start off very simply. So if you were going to draw a picture of the bull in the upper left-hand corner, an artist would start off with the minimal elements of the bull, which is the picture in the lower right-hand corner. And over time, you can add complexity to this bull, but you have to start off with something simple. Next slide. So we created a simple model looking at breast cancer and looking to see how is it that breast cancer spreads from one organ site to another. And the person we got to do this was not a biologist, was not a physicist. This was a mathematician who predicts weather. And what we wanted to do was apply the techniques of weather prediction to cancer. Because of the question I get asked all the time by my patients is, Dr. Nieva, is my cancer going to spread to my brain? And my current answer is, I have no idea. There's some probability that that will occur. You say, yes, but what is that probability? Well, let me get out my model, and let me calculate that for you. And we developed this very simplified model of the human body that identified the paths that cancer takes from one site to another. And so to answer my patient's question, is my cancer going to spread to my brain? The question I need to ask first is, where is your cancer now? Next slide. And so we analyzed over 400 patients from a cancer center in New York and looked at the patterns of spread and found that breast cancer most commonly goes to the bone. Most commonly after that is the chest wall, then the lungs, then distant lymph nodes, then liver, and then other places. But once it's gone to the bone, the next most likely place that it will show up is the lung. But if it first goes to the liver, the likelihood that it's going to go to the lung is actually much lower. It's much more likely to go to a lymph node or to bone or just stay in the liver after that. So we can start to make predictions based on where the cancer is, and that's what this tree ring diagram of our patient shows us. Next slide. So this is another example. This is a young man I met this last year, and he has an interesting company and an interesting technology. He's an engineer who did his PhD in Australia. And what he can do is he can get a new gene into a cell. And he can do that without using a virus, without using any other technology, simply by flowing the cell through a tube under the right pressure conditions and diameter of the tube. And so he made these microchips that are capable of doing that and modifying cells and getting genes into cells. And he described his technology to me, and I said, that looks great, but the best solution for you and the best application of your technology is not the problem I work on. It's not lung cancer. It's not head and neck cancer. I told him the best place he could apply his technology was in a disease called hemophilia to try to create stable gene transcripts. And so that's an example of the people who are in these relationships, these convergent relationships. It has to not always be about their problem. I had a conversation yesterday with a friend of mine who had a new technology, and the new technology was potentially useful in prostate cancer. And it seemed like every physician he talked to said, well, why don't you apply your technology to prostate cancer patients who have a rising PSA but don't have any disease? And he said, yeah, that's a great idea, except the FDA would never approve that particular indication. It would take over 20 years to demonstrate that it was beneficial in that subgroup. So I asked him, why are the doctors giving you bad advice? Why are they telling you to go into a disease area that doesn't support your technology? And he said, well, it's not really helpful to my technology to develop a clinical trial in that area, but that's where those doctors want clinical trials. And I think the lesson from most convergent technologies, if they're going to be successful, is everybody has to go into it thinking not about their own particular niche, about their own particular best interests, but what can the technology do? How will it best help patients? And maybe it's the patients I treat, but maybe it's not. And that was, and this example is one where it's going to help patients, but probably not the ones I treat. Next slide. So I met a mathematician and a computer scientist, and recently he showed me his data on multi-scale time series analysis. And basically, he could look at data points in 3 dimensions, in 4 dimensions, in multiple dimensions, and as long as they were on the same time series, he could analyze what was happening to someone. And he actually had a problem he was working on. He was going to use the images of a human from a Microsoft Kinect video game system to diagnose people with Parkinson's disease. I said, gee, that's terrific, but really what I need, and my problem is I need my performance status on my patients evaluated in an objective way. He said, oh, that'd be easy. And so we've now created this team, this project called the Atom HP Project, and this is with Cyrus Shahabi at the Viterbi School of Engineering. And he's a guy who's used to analyzing things like traffic patterns just through mathematics and computer science. And what we're going to do is we're going to take pictures of our patients, video actually, using a Microsoft Kinect. We're going to play some Microsoft Band on them to track their movements. And we're going to get very careful measurements and objective measurements of their performance status and activity. And one could imagine that this could be used in the future for patients who are receiving chemotherapy. And if they start not doing well at home, like they don't get out of bed for 2 days, this type of device could monitor and send an alert to the physician that there may be a problem. Next slide. So as Carol told you, this is the kind of stuff we do in Convergent Science, which is a journal we're very excited about. Next slide. And love to open it up to questions and get people's thoughts on what we described.
So this is Peggy, and I want to thank both of you. I think we had about 50 slides or so, and that I've got about 50 wows. Every slide had something interesting and amazing on it. So thank you to both of you. While questions are getting typed in, so if you have a question, please type them in. I've got a few for you, Dr. Nieva, and also for you, Carol. One of my questions is, so I love the clones and being able to capture the cells and being able to characterize them and learn new information from it. And I'm wondering how this whole, if you can do this liquid biopsy pre-chemo and then post-chemo and measure the differences and see what happens there.
Yeah, that's exactly what pharma's doing. We don't do this type of stuff in the academic labs. We've actually kind of farmed that out to a commercial lab that now does it, and that lab is called Epic Sciences, and it's based out of San Diego. And what it's being used for now primarily is as a drug development tool. Basically, as the pharma companies are developing a new drug, they want to know, did my drug shut off the target in the cells that I'm interested in? So what they'll want to do is take the fluid biopsy, give the drug, do another one, look to see if the drug shut off the protein, the target, the signaling pathway that was intended. This is particularly important at the earliest stage of drug discovery, the phase 1 phase, when they're gradually escalating the doses and they're trying to find the right dose of the drug that's going to have an effect. So yes, this is being done. This is being done by pharma primarily as a development tool. It's not yet something that is being done as a patient sort of clinical device. We're not having patients, for example, go get their Taxol and then see if the microtubules in their mitotic spindles have been frozen. That's not where the technology is. It's still in that research phase.
Great, thank you. That's amazing, and I could see it in the clinic one of these days, hopefully. So the second question I have is this whole seed and soil. You've got these circulating cells. Is anyone looking at the microenvironment to see how that interplays with? We had a big discussion a couple talks back on, okay, so you have these circulating cells, but do you have to have the right environment for them to attach to begin growing and forming, metastases from?
Yeah, I think we do. We know that certain patients are more likely to have these circulating tumor cells than others. And some of that may depend on the soil at which the circulating tumor cells are coming from. And we think that certain areas, particularly the bone marrow, may be a looser soil. And we may be seeing more circulating tumor cells coming out of it. From the standpoint of interactions between the cells and the soil, we're opening a clinical trial here at USC where we're going to try to disrupt that interaction between two genes. One is called FB2 and the other one is FRNB4, or actually FB4 and FRNB2, and that interaction between the tumor cell and its vascular supply is a very powerful protein-protein interaction. So actually, Peter Kuhn actually first crystallized FRNB4, demonstrated its structure, and then we have Prakash Gill here at USC who engineered a molecule that could disrupt that interaction between the tumor cell and the soil to basically try to uproot the cells. And we'll be looking at correlates and circulating tumor cells in that particular trial. We'll probably be opening that trial in the summertime.
Great, thank you. We have a question here from Stanford James. So, Stanford, I've just unmuted you if you'd like to ask it.
Can you all hear me?
Yes.
Oh, great. So I did post the question, and I think, by the way, Carol and Dr. Webb, I think it's a very good presentation that both of y'all gave. And I'm always curious, and I'm curious to know how we can, as advocates especially, how can we improve the knowledge and awareness of this type of work in our roles as advocates? And I'm being reminded of existing state cancer control plans, which many of them address the need to include research as part of the cancer plan. And it would appear to me that this type of work is going to be an improvement in not only the knowledge and awareness of certain cancers, particularly around cancer in minorities. And I'd be curious to know if there's an effort to really collaborate with, state cancer agencies and state cancer plans for this reason. It would seem to me that advocates can really help convey the message and mission because there's so much great work being done, and Carol alluded to it too. Much of this work, particularly around physics, is unknown, and so therefore it's great if you're a physicist, it's great if you're, even if you're a scientist, but many patients in many areas don't go to an academic center for their treatment. So it would seem that as advocates who are more of a local level, if we can think about, okay, is there a desire to convey the research that you guys are doing so it can coincide with the desire of many state cancer plans, which is supposed to include research. It's supposed to include research to improve the cancer disparities and mortality levels. So I hope I had 3 questions, but that's the main thing is about how can we as advocates really work hard to convey this work to other people. And it would seem to me the state cancer plans, which every state has, it would seem that that's a way that might can be done because much of the work is unknown beyond certain areas.
So, Carol, do you want to take the first shot at that?
Okay. It's been hard to get advocates involved in this space. We have to get the word out about this type of research. One of the problems that I've had is when I communicate to patient groups, they're usually much more excited about clinical trials and things that perhaps are more easily understandable to them. But what I've tried to do over the last 6 years or so is just to really spread the word about this type of cancer research and explain to people the importance of it. So the journal, I think, is really going to be a good step in communicating and kind of opening up this information to the public. You know, people, if they know it's out there, if they understand the importance of it, I really think that they'll be excited. You know, it's kind of like science fiction in a way. So the journal's going to explain in lay language some of these very complicated technical approaches. It's going to be open to readership for advocates. Now, in regards to your comment about, you know, the state cancer control programs, I don't really know how we can work with those programs other than a lot of I mean, I came from an engineering program, right? There's a lot of schools that teach engineering that really could work in this space, and how can we tie that in with state cancer programs? I'm not sure. It's probably going to take a coordinated effort by a larger entity, you know, the NCI perhaps, and I'd love to see that happen, but I just don't know how they'd go about it. So anyway, Jorge, you want to take a crack at it?
If I could address the issue of how the physical sciences help with cancer disparities, because I think that's a very important issue. One of the things that these mathematical approaches and these computational approaches to diagnosis do is they sort of deconvolute the complexity of what's happening to patients. They make it so you don't have to go see the most brilliant clinician in the world to get the right diagnosis. And you don't need to necessarily be at a place that has a lot of resources. We've submitted grants for this fluid biopsy technology to be applied in second and low and middle income countries, places like China, Mexico, places that don't have a lot of resources, because the technology is really very cheap. Computers are not expensive. Computational technologies are not expensive. And they give us an opportunity to democratize some of the very complex things that happen at academic medical centers and really bring them to people who are being treated at community centers. So, you know, for example, this technology that lets you put into a little calculator whether or not somebody's got lung cancer on the basis of a couple of little calculations, you know, these are very powerful tools for patient counseling, and they're very powerful tools that are easy to understand regardless of one's level of education, regardless of whether one is in a high or low resource area. So I think actually the disparities part is actually very important for looking at these technologies. Whether or not the states can help us, you know, I've never written a grant to a state. I've never seen a grant offered by states that help in my research. So I have trouble sort of understanding, you know, what the role of states are in supporting medical research. For me, it's been my entire career. I've really relied on federal funding and private funding. There's state funding out there. I'd love to hear about it.
Stanford, did you need to say anything else? If you're finished, I'll mute you.
The only thing I was going to address, particularly around the whole thing about disparities, and I'm really glad that you addressed that, because it would seem to me, and I had a chance to meet with Peter Kuhn, and he is an amazing scientist, and his desire to really engage around disparities is really welcoming. And I did want to say that, and in answer to what you were saying and also what Carol was saying, it would seem to me that through organizations like the NCI, there is an avenue. Many of these state cancer control plans are funded by the CDC, which in part gets their money through NIH, there's NCI. There is a stream of connection, and as an advocate, I can tell you right now, there's also a lack of communications network with all of these agencies, particularly from the state level. So, I think that, again, as an advocate, myself, I'm connected to different groups, but the problem we run into, even as you said it, there is very little funding to address the health disparities and the work that's done more of a community-based nature. Much of the funding will go to the national organizations, and they have a disconnect with local. So the only thing I wanted to say was, I think there is an avenue coming from the NCI to actually do it, but we first have to acknowledge the fact that it is not being done. So thank you very much.
Thank you so much. I'm going to mute you again because there's a little bit of interference. But
thank you.
Write another question if you've got one, Stanford. I'm going to unmute Jim O'Mell now. Okay, Jim, go ahead.
Hi, Peggy. Are you there?
Yes.
My question was, can circulating tumor cell evaluation replace traditional biopsies?
I think it can. And I would actually like to see greater efforts on the part of pharma. I mean, maybe you saw the article that was in the paper, just a couple of days ago. It was basically the result of a research trial that showed that we've put such a focus on biomarker discovery in our cancer clinical trials that we're now almost universally requiring biopsies for people to enroll in cancer clinical trials. And some of these biopsies are not only of moderate risk, but also the biopsies sometimes preclude patients from enrolling. I've had patients who couldn't enroll in a clinical trial because their tumor wasn't in a place where you could do a core biopsy. It's because I think the NCI has put a big focus on biomarker discovery, which is great. Pharma's recognized the importance of biomarkers, which is great. The FDA has said you must have a biomarker, and that's great. But I really think we need to get back to a risk-benefit calculation as to what we're getting out of these biomarkers. And I think fluid biopsies are a great way to, as I like to say, democratize the process so that, somebody can enroll in a clinical trial even if they're at a center whose interventional radiologists refuse to do a core biopsy in the lung, which a lot of interventional radiologists don't like to do because it bleeds a lot. And they said, no, we'll only put a little tiny needle in the lung. Well, that doesn't cut it for my clinical trial that's sponsored by Stand Up to Cancer. They need a big biopsy. And so I would like to see greater efforts developed at recognizing that you can get a lot of these biomarkers from the circulating tumor cells, and a recognition that not only can you do it, but we probably should be doing it. But currently, that's not the current zeitgeist. But I hate seeing patients not being able to go on trial because they can't get a biopsy.
It seems that if it's possible, that it should be done. It just seems intuitively the way to do it.
Our regulators like to set high bars for things. And they say, no, we want to be perfect. We want to have great science. We want to have great biopsies, big biopsies, validated biopsies that we can distribute to lots of scientists. That's great. Wonderful to set a high bar. But when you set that high bar, there are patients who fall below that bar, and then they don't get in. And I'd like to see the bar set a little lower where we're doing more of these fluid biopsies, are they as good as real biopsies, tissue biopsies? Maybe not. You lose some information. You lose microenvironment information, for example. You lose a lot of information about the interactions of the immune system with the cancers. But I think it should be an option for a lot of people. And they should have that ability to say, well, if we can't jump over this bar for the purpose of biomarker discovery, we can instead jump over this bar.
I'd also sent a suggestion to Peggy about. I had also sent a suggestion to Peggy about your trial in which you release cancer cells from their soil. The suggestion is to plan on timed systemic therapy to be given to those patients shortly after that cell release. It just seems like so many cancers have niches, specifically myeloma in the bone marrow, where the cancer cells are so hard to kill that if they could be released and then give systemic therapy, the timing of that therapy, it would just seem like a very appropriate treatment.
And we're actually doing that trial combining the Ephrin B4 agent with chemotherapy, and we're doing that in lung and head and neck cancer. The clinical trial that's going to be opening is going to be doing it with immunotherapy. So yeah, I'm completely with you.
Thank you.
If there's any other questions, keep typing. While you're typing, I've got maybe I don't know whether it's a suggestion or an ask. Maybe it's a big ask. If Carol, if you and Dr. Yeva can speak with the mathematicians that you're involved with. I know this is not a very exciting area, but for patients, it's a really important area that all the clinical trial data be converted from relative risk numbers to absolute risk numbers, that almost everything we hear is in a relative risk number, and a 30% that's really an absolute risk of maybe 4 or 5% is a big difference for patients. It's really hard to make a decision when you're getting relative risk numbers. And if you can get these mathematicians to go back, I don't know, to some of the old trials and gather the data, or even go moving forward from now on, just everything get reported in absolute numbers as well. It's just something that's driven me crazy for years.
You know, that's your doctor's job, and your doctor should be able to actually do those calculations in their head. They all had good SAT scores on the math SAT, and they can do that. They can do that arithmetic. People know what the absolute risks are. If they don't know what the absolute risks are, you know, there's great pieces of software out there and models such as Adjuvant Online, which is used in the breast cancer field very frequently, where absolute risks are calculated. And I think one of the things we're often guilty of in medicine is we're guilty of over-treating diseases. You know, we do a lot of treatment of hypertension, we do a lot of treatment of cholesterol, we do a lot of treatment to reduce risks. We put patients on a lot of medicines to reduce risks. And we do that often on the basis of these relatively high relative risks, which when you go back and you calculate the absolute risks, they're really, really quite low. You know, I saw a patient once a couple of weeks ago. He was a cancer patient of mine, and he got a heart attack. And I said, well, are they going to put him on aspirin? They said, yes. Oh, and they're also putting him on Plavix. And I had to ask my resident, well, what's the absolute benefit of adding Plavix to aspirin in somebody with advanced cirrhosis? And we, you know, you go out, you pull the papers, you do the calculation. Well, gee, it's about a 0.5% benefit to add that extra medicine. But that extra medicine was going to increase the bleeding rate in this patient by 3 or 4%. So I'm entirely with you on the issue of absolute risk. I wish that all of my colleagues would only talk to patients in the sense of absolute risk for just about everything we do. I think we'd find that patients would take less treatment if they really knew those numbers.
Right. I mean, just an example in the news a couple weeks ago was, you know, eating processed meats and burnt hot dogs and all of that. And I can't remember whether it was 25 or 28% or something. Your colon cancer risk went up. But I happened to be at an AACR meeting sitting across from a colon cancer researcher, and I said, what's your lifetime risk of getting colon cancer? And he said about 5%. So, you know, it's not 28%, it's, you know, 28% of 5%. There's, you know, so it's a very small number, but the news just made it sound like, whoa, you're crazy if you do this. And so just anytime you can talk to any of your colleagues about that, because everybody I encounter is giving relative risk numbers, not absolute risk numbers. So one other question I have while we're waiting for more questions to come in. So people type your questions in. We're running out of time. So one of the ones, Dr. Nieva, you mentioned some of these new tools to measure patients and their movement and all of this. And I'm wondering how that would tie into the patient-reported outcomes, because that's becoming a really big field. And yet if you can merge it with actual mathematical data or, you know, engineering data where you actually see what's happening and then hear what they're saying. I'm always interested in what people say versus what's actually happening.
That actually is a big part of that project. And Arthur Stone and Joan Broderick, who are here at USC with me now, they came over from Stony Brook, and they're the ones who developed the NIH PROMIS patient-reported outcome measure. They're participating with us on this study of wearable monitors, and we're going to have patients do a patient-reported outcome actually on a daily basis while we analyze their movements, because I think it's important to know, do we need to monitor patients' movements? Some patients may not want that. Is it just as good for them to just answer a question on their smartphone every day? That might actually be cheaper, that might be easier. So we're very interested in that question because we want to know also how other aspects of life affect movement beyond the physical. So we're asking not just about things like physical fatigue, we're also asking about things like social isolation, which I think will may have an effect on how much patients move about in their daily life.
Tremendous. Great. Super. So Stanford has a few more questions.
Okay, I have lots of questions, but I'm going to ask one. What has been the outcomes of the research completed so far, especially around even the work with CTCs and other things? And what has been the outcomes, and has there been any effort to have this research outcome transfer to change in clinical practice?
Sure. So the CTC technologies continue to be a work in progress. The first way that they were transferred to outcomes is the technology was licensed by a corporation that is now hired by industry and has become a routine part of drug development. So our thought is that it is reducing costs for developing new drugs by evaluating them better and better understanding what dose are they working at, whether they're hitting their targets at early doses and later doses, and the hope is that that's leading to better understanding by pharma as to what is the right dose of a medicine to take to the next level of clinical trial. And I think it works because pharma continues to be a repeat customer of that company, and that company's gone from like 5 employees like 5 years ago to over 100 employees. So they're busy, they're doing work, and pharma keeps coming back and giving them more work. So that's been an outcome that's become a practical solution. From the standpoint of other outcomes, it's been primarily research publications and then continuing to drive the field forward. You know, when we started doing this work, the term fluid biopsy didn't exist. that was that was our sort of word to describe what it was that we were doing. And I now use fluid biopsies in the clinic, and I do them routinely. That's become kind of an integrated part of my practice for patients who can't get tissue biopsies done. Now, the fluid biopsies I use aren't the ones that we do in the lab. I use circulating free DNA assays primarily. But the concept is the same. And what we want to get to is kind of merging those two things, merging genomic analysis that you can get from plasma with cellular analysis that you can only get from cells. So essentially, we're just kind of continuing to build on what's happened in the past. What was the second question? Because there were two there. You still there, Stanford?
I'm still here, and I didn't know Peggy was just going to unmute me. So my question is, and I really like what you just said, and I'm going to stop asking questions now, but I get curious. I'm really curious to know, is there an effort to utilize social media as a way of conveying this a little bit better? I only ask that because this is such a great medium that many advocates are utilizing, but also all of these agencies, from state agencies to federal, they all have a social media presence. International organizations, from AACR to ASCO to everyone else. The only question I have is, it would seem that there is a way to collaborate better by utilizing these existing networks, which is why I first mentioned those cancer control plans, which are on the ground looking for strategies. They have strategic plans and goals, but they don't have a way of actually implementing any of the strategies. That's what they're looking for.
So, we do a little bit of social media. We have a Facebook page, the Cancer Fluid Biopsy Research Group. You can like us on Facebook. I'm probably not as good at social media as most of the people who are on this call. So maybe Carol, she can help me. She's, I think, better at social media than I am.
I think this is a really great area where advocates can help us. We tweet from the journal, for example. I find Facebook is a great way to spread information, but it's just right now. I think it behooves all of us in the advocacy community to know about the different types of research that are out there and to really spread the word. Now, I'm not I know that some other advocates are much more connected in social media than I am, and it would be awesome if we could collaborate, with those advocates and figure out a way to really make what you've mentioned happen. So, and on a different note, Peggy, I can't type in my questions, but I have questions. So when there's a space in the queue, my hand is raised.
Okay, Stanford, are you good?
I'm good. Thank you so much, everyone.
Okay. Go ahead, Carol.
Okay. I know, Jorge, that you have been on papers that talk about, use the term sponges and spreaders. And some of the advocates on this call may have heard that terminology before. And while you're here, I was wondering if you could kind of explain that whole concept to us.
Sure. Well, there was a discovery that came out of a mouse experiment, and I very rarely quote mouse experiments, but this was a good one, where they took fluorescently labeled cancer cells, put them in one flank of the mouse that were green, and in the other flank of the mouse they put in yellow cancer cells. And then they let the tumors grow in each flank of the mouse. And what they found was that if you then went back and analyzed the fluorescent labels on the cancer cells in those mice, they were mixed. Basically, the tumor had spread from one cancer site to the other, and there was free trafficking of cancer cells from one site to the next. And the same thing happens in people. And what these Markov spreader sponge diagrams demonstrate is that the cancer cells don't just go in one direction. It's not a tumor that starts in the breast, goes to bone, and then goes from breast to lung, and then breast to liver, and so if you just took the breast out, everything would be fine. The cancer actually crisscrosses and traffics in the circulation and goes from one site to the other. And that may be why cancers, instead of growing like balls, that may be why they grow like snowflakes, which is the same way that snowflakes kind of grow. They grow from the things being added to the outside. So once you have that understanding and you have that recognition that cancer doesn't just grow in place, but actually traffics back and forth, you can then start to ask questions, well, is the rate of trafficking back and forth the same from all sites? Is the rate of cancer cells coming in and the rate of cancer cells going out the same everywhere? And it's not. And the models demonstrate this, and experiments have demonstrated it as well. There are some sites where the rate of cells going out is faster than the rate coming in, and there are some sites where the rate coming in is faster than the rate going out, and that's what we describe as spreaders versus sponges. Spreaders have a net outflow of cancer cells, where sponges have a net inflow of cancer cells. Now, all sites have growth, all sites have cell division happening in them, but there is also this trafficking pattern. And so what these Markov models show is that in breast cancer, for example, the bone, the chest wall, and the lymph nodes, as shown in panel A, are spreader sites. They tend to send cells out more than coming in, whereas the lung, pleura, and liver are the opposite. And the interesting thing is that not all subtypes of breast cancer are the same. So if you look at those Markov diagrams in panels B, C, and D, what you see is that for the luminal type breast cancers and for the triple negative cancers, they're a little different in terms of their patterns of spreaders versus sponge. And for example, in the triple negative breast cancer population, the order at which those different nodes are centered around the circle is different than it is for some others. You see that spread locally to mammary lymph nodes happens early and is very prominent in the triple negative subtype, whereas spread to liver is less common, whereas spread to liver is much more common, occurring earlier in the sequence, because those nodes are organized like a clock face. And the closer you are to the top of the clock, the more important that particular node is. Liver becomes much more important in the HER2 positive setting. You'll also notice that in the HER2 positive setting, the lung converts from a sponge site and converts to a spreader site. And so we see fundamentally different biology in the HER2 patients with regard to their lung metastases than we see in the other populations.
Thank you very much.
Do we have more questions, Carol?
How does, I know that increases our understanding of cancer. Has it gotten to the point where it really affects the patients in the clinic? Are we there yet?
When we made these diagrams in lung cancer, one of the things we identified was that unlike all the other cancers, when you build these spreader sponge diagrams, all the other cancers typically spread from the primary site to lymph nodes being very prominent. And what we found in lung cancer was that for many patients, the adrenal gland was actually a more prominent first site than the lymph nodes were. And there had always been a little bit of literature out there suggesting that patients who had lung cancer metastasized to their adrenal glands could benefit from surgery. But the analysis of our autopsy series really reinforced that finding. And the model really reinforced that finding of the importance of the adrenal glands. And so we've actually had a site in the UK say, oh yeah, saw your work on this. We're now taking out the adrenal gland solitary metastases routinely. And that's just what we do now. So in a way, it has affected patient care. That's a very small niche. But I think those are the types of small niches where we can, I think, bring attention and recognition to something that people may not have been thinking about before because it didn't really jump out of the noise of all the other data before.
Okay, that's wonderful. And I just wanted to tell everyone that I was so happy that Dr. Nieva could join us on this call, because one of the things that I really wanted to point out was that clinical oncologists are working in conjunction with the physical scientist, and it does affect patients. And we had a lovely example, here of how. So thank you.
My privilege.
So Carol, I have a question